there are some ways to avoid superinfections in cases where you have to be put on broad-spectrum antibiotics, because those are simply the only antibiotics that are useful to treat the particular infection that you have. And that is the use of probiotics and prebiotics. I mean, really, probiotics and prebiotics should be something that are used daily to help colonize your normal biota and maintain gut health. But if you're not doing them daily, at least try to implement their use when you are taking antibiotics. So probiotics are actually ingesting healthy living microorganisms. So this can happen in a variety of ways. It could be something as simple as eating a yogurt. If you look at the back of a yogurt and look at the ingredients, it will either say live cultures, or it will give you a few of the cultures that are in it, such as lactobacilli. But you can also take probiotic supplements. There's powders and vitamins and all different types of ways to get probiotics nowadays. So again, by ingesting probiotics, you are putting good, healthy bacteria into your system, especially your gastrointestinal system, to help maintain your gut normal biota and your gut health. Prebiotics are different. Prebiotics are ingesting nutrients that will allow the normal biota that you already have in your system to grow faster. A lot of fruits and vegetables are natural prebiotics, so when you eat those, it just stimulates the microorganisms, the good microorganisms that you have in your gut, just to grow and divide faster. So they're different from probiotics. Probiotics are actually ingesting living, healthy microorganisms, whereas prebiotics are just ingesting food that is going to help support the normal biota that are already present. In extreme cases, where patients have been on broad-spectrum antibiotics for a period of time and have recurrent bouts of C. diff, fecal transplants are utilized. So again, this only happens in specific cases where, again, people are continuously getting superinfections such as C. diff because they have to repeatedly be on broad-spectrum antibiotics. So it is what the name says. They typically will get a fecal transplant. So donated feces will be transferred into their gastrointestinal system via colonoscopy. It's not as disgusting as it sounds. The feces is filtered from out of any type of impurities or anything potentially toxic. So really what you're getting from the fecal transplant through the colonoscopy is the healthy gut bacteria. So that healthy bacteria is going to help colonize the gastrointestinal system and the intestines much faster than something as simple as taking probiotics. So it is a kind of a newer-ish technique that's being utilized, and while it's not, utilized for every different case of kind of normal biota depletion, it's definitely beneficial in cases where patients have recurrent bouts of C. diff. So the next thing we'll talk about are modes of action. So every antibiotic has a mode of action, which is basically how that antibiotic inhibits the bacterial cell from growing. Does it target the cell wall? Does it target the cell membrane? Is it stopping protein synthesis? Is it preventing DNA or RNA from being produced? Is it stopping some type of metabolic reaction from happening? So really the way antibiotics work is they target part of a bacterial cell, and that prevents bacterial cell growth. Ultimately, the bacterial cells will die, and then that obviously can be used to cure an infection. So in this course, we don't have time to go over every single antibiotic, although if you're going into one of the healthcare programs, you will take a pharmacology course that goes over the antibiotics. But for this course, what I've done is pull one antibiotic out of each category. So that way, if you know how that antibiotic works, you know how all the other antibiotics work within the same category. So we are going to be looking at specific antibiotics and seeing what part of the bacterial cell they target in order to stop bacterial growth and ultimately cure an infection. So the first are antibiotics that target the cell wall. So antibiotics such as penicillin and vancomycin, they work by destroying peptidoglycan in the cell walls of bacteria. So these antibiotics have a chemical structure called a beta-lactam ring that essentially acts like a scissor, and it cuts the peptidoglycan in the cell wall of the bacteria. And with bacteria, their cell walls are very important. It helps maintain their shape, structure, and support. So if it gets, harmed in any way, the bacterial cells will typically burst through cell lysis. And that's exactly what penicillin is going to do. It's going to use its beta-lactam ring to cut the peptidoglycan in the cell wall, and then ultimately the bacterial cells will burst through cell lysis, and that's going to treat the infection. So up here we have pictures of bacteria going through division. So this is before they've been exposed to penicillin. So they're happy and they're growing and dividing. We can see them going through binary fission cycles. Down here, they've been exposed to penicillin. So penicillin went in with that beta-lactam ring and cut the peptidoglycan in their cell walls. And we can see how they are basically expanding and bursting through cell lysis. Unfortunately, a lot of bacteria have gained resistance to penicillin. And the reason they're able to do that is through the action of this enzyme called beta-lactamase. If a bacterium has beta-lactamase, it is able to destroy the beta-lactam ring in penicillin, which will render penicillin completely ineffective. So if you are infected with a bacterium that has beta-lactamase and you take penicillin, penicillin's not going to be able to do anything because the bacteria can destroy penicillin before penicillin can target them. So beta-lactamase is a very common enzyme among bacteria, and that's why we see so much resistance towards penicillin. Polymyxin is an example of an antibiotic that's going to target the cell membrane. So you don't want to get the cell wall confused with the cell membrane. They're two different layers. So penicillin would be an example of an antibiotic that would target the cell wall and the peptidoglycan in the cell wall. Polymyxin targets the phospholipids in the cell membrane, which again, the cell membrane is the layer that's responsible for selective permeability or transport of things into and outside of the cell. So what polymyxin does is it basically binds to the cell membrane and basically destroys some of the phospholipids in the cell membrane, so kind of pokes holes in the cell membrane, and then ultimately the internal contents of the bacterial cells start to leak out and the bacterial cell shrivels and dies. So polymyxin destroys phospholipids in the cell membrane, and then ultimately bacterial cells shrivel and die once they're exposed to polymyxin. Quite a few antibiotics target protein synthesis. So with these antibiotics, ultimately they stop bacterial cells from making proteins. And if a bacterial cell cannot produce proteins anymore, it can no longer grow and divide and it's going to die off. So we have 4 different types of antibiotics that target protein synthesis: streptomycin, tetracycline, erythromycin, and chloramphenicol. So it's not enough just to say that they stop protein synthesis. We want to know exactly how they do that. So if we remember from previous lessons, the ribosome is what's responsible for protein synthesis. It reads the messenger RNA and then builds the chain of amino acids based on the code provided by the messenger RNA. We know that the ribosome has 2 subunits, and in bacteria, the large subunit is 50S in size, and the small subunit is 30S in size. So antibiotics work by targeting a specific ribosomal subunit and effectively inhibiting protein synthesis. So streptomycin and tetracycline, they work by binding to and inhibiting the 30S ribosomal subunit. But ultimately, that stops protein synthesis or translation, and the bacterial cells die. Erythromycin and chloramphenicol, they do the same thing, but they bind to the 50S, the large ribosomal subunit. But again, by binding to the large ribosomal subunit, they stop protein synthesis or translation, and the bacterial cells die. It is important to note that while humans do have ribosomes, our ribosomes are bigger. Our large subunit is 60S, our small subunit is 40S. So these particular antibiotics, while they do target ribosomes, they are not going to bind to and destroy our ribosomes because they wouldn't recognize the size of our ribosomes. They are designed to target specifically bacterial ribosomes, which are 50S and 30S as far as their subunits. Then there are antibiotics that target nucleic acid synthesis. So if you remember from our last lesson, there are two types of nucleic acids. There are DNA and RNA. So there are antibiotics that target the synthesis of RNA, which we refer to as transcription. And then there are antibiotics that target DNA replication, which is the synthesis of DNA. So an example of an antibiotic that targets RNA synthesis would be rifapin. Rifapin binds to the RNA polymerase enzyme, which is the enzyme that's used to make RNA during transcription, and it stops it from working. If a bacterial cell can no longer make RNA during transcription, then it's eventually not going to be able to make protein during translation, and ultimately the bacterial cell dies. So rifapin inhibits the RNA polymerase enzyme and basically inhibits transcription or RNA synthesis. Ciprofloxacin is an antibiotic that's going to inhibit DNA replication, so it prevents bacterial cells from replicating their DNA to pass on to future progeny, new bacterial cells. Specifically, how the antibiotic does this is it inhibits the action of the DNA gyrase enzyme. This enzyme is necessary to unwind the helical double-stranded DNA molecule. It's basically responsible typically for the supercoiling that's used to coil the chromosome in order to package it. So this enzyme is required in order to uncoil for replication. So if a patient is taking Cipro, it's going to prevent the DNA gyrase enzyme from working. And the DNA is not going to be able to uncoil, which means it's not going to be able to go through replication, and the bacterial cells are not going to be able to produce more of themselves, which ultimately is going to cure the infection. So Cipro is responsible for inhibiting DNA replication by preventing the DNA gyrase enzyme from working. And the last antibiotic we'll talk about are the sulfa drugs such as trimethoprim. This is an antibiotic that prevents the production of folic acid. Folic acid is necessary for bacteria. They need it in order to engage in DNA replication and protein synthesis. So it is a mandatory nutrient for them. So if you're taking trimethoprim, it basically blocks bacteria from producing folic acid, so they would no longer be able to engage in replication and translation, and then ultimately the bacterial cells would die. It is important to note folic acid is important for bacteria to support growth through DNA replication and protein synthesis, but folic acid is also important in humans, again, to support growth. It's especially important for pregnant women. The main ingredient in prenatal vitamins is folic acid. So pregnant women need to have a higher concentration of folic acid in order to maintain growth and development, obviously because they're making cells and tissues for the fetus. So you would never ever want to get a pregnant woman trimethoprim because it can decrease the folic acid levels, and that could ultimately affect the fetus. So while trimethoprim is really great if you're not pregnant to inhibit bacterial growth, it is not a good medication to give to someone that is pregnant if they have an infection.